Hello, everyone, and thank you for joining us for another special episode of the Animal Care Systems webinar series. I'm your host, Austin Carell. We again want to thank ALAS for helping to distribute the webinar series through the ACE community, and we thank the other participating companies. Today's presenter is Ms. Susan Freling. Ms. Freling has been a veterinary technician for more than 25 years with experience in all facets of veterinary. Ms. Freling helps manage the tree shrew colony at the Max Planck Institute for Neuroscience in Florida. She helped establish the colony and develop new strategies for enrichment, raising infants, behavioral training, and dietary needs. Ms. Freling also is part of a research team that uses tree shrews as a model for the visual system. Animal Care Systems is delighted to host Ms. Freeling's webinar today. The title of her seminar is, I didn't pick the tree shrew life, the tree shrew life picked me. If you have a question for Ms. Freling, please use the question pane in the control panel, and we will answer as many questions as we can afterward. And now please welcome Ms. Susan Freling.
Hi, everybody. Thank you so much for having me, Austin. And he definitely is, he's the guru for this in this crazy world that we're living in right now. Be patient as far as if we have any technical difficulties. Trust me, it's on my end, not on his. And if you happen to see or happen to hear or see my dog or cats roaming around, dinner's a couple hours away. So they're circling me like buzzards right now so they don't miss anything. Anyway, thanks everybody, and let's get started. Okay, so as Austin gave you a little bit about my background, I'll tell you a little bit about our group that we have at MPFI. Early on, there were three of us, three main people that were taking care of the shrews and learning about them and trying to give them the best life that we could. And those three people obviously are still here today, thank goodness. But between the three of us, we had a wealth of information and knowledge that we brought to the table. As he said, we had many years of vet tech experience. We also had zoo experience, wildlife experience, and this really, really has become critical in helping us to better understand these little guys for sure. So the first thing that I want to say is everybody obviously doesn't know what a tree shrew is. More do now than they used to, but they see those crazy little teeth. And the first thing a lot of people think about is Scrat from Ice Age. Obviously, it's not him, but hey, you never know. He could be a close relative. So let's do a little bit of background history. Let's go with what exactly is a tree shrew. So they are known as Tupaia Belangeri, the Northern Tree Shrew. That's what we have at Max Planck, but there are many more species, actually about 20 that we know of right now. Despite their name, they really don't live in trees, and they actually aren't shrews at all, as we would understand them. They do forage on the ground, so you will see them up in the trees, obviously, to navigate around, but for all intents and purposes, they do not live in the trees. They're originally from the tropical forests of Southeast Asia, and the first report was a long time ago. They were originally designated as squirrels, of course, because they look like a squirrel for the most part. And over the years, many years, it progressed back and forth. It was rodents, are they primates? You know, really, where do we go with this? Finally, somebody said, you know what? We have no idea because we see a lot of different things from different animals that's similar to these guys. So let's just put them in their own order called Scandentia. Their average lifespan in the wild is about 3 to 5 years, and in captivity about 8 to 10. They are active during the daytime, and their body weight ranges from about 120 grams to 270. I would say a nice average, and this is male or female, is probably around that 180 range. They are insectivores, so in the wild they're going to be eating those really small mammals, you know, maybe mice or things like that, insects, leaves, fruit. We feed them insectivore diet. They have a rotation of vegetables and fruits, mealworms, things like that, some crickets, and on top they also get a high quality cat food as well. They do communicate with scent marking and vocalization. They have a big scent gland on their neck, throat area that they will mark all over everything. And then they do, of course, see color very well. They recognize people, even the little guys, even the juveniles, you know, a couple of months old. They do have 8 distinct vocalizations that have been studied. There are many variations of it, but there are 8 specific ones. And actually, they have the highest ratio of brain to body mass of any mammal. So for a general understanding of the species, we'll start with that. So where do you find the info? If you don't know, you know, what's going on and how do you figure it out? We do have a little bit of information, obviously, that's in the textbooks. We do have a lot of scientific papers in the past several years that have been written, which has helped us tremendously. But for that general information, we really have to work together and all of our collaborators to see what that normal is. Luckily, we've had the shrews for 9 years now, so we've gone through those hardships, so to speak, and the joys, of course, but in trying to figure out and just learn from the animals exactly what is normal for them. As with anything, you're going to look for the posturing, of course, the food, water intake, feces, urine, anything like that, and just how they interact with not only you, but of course their cage mates. We do, of course, our training in-house when we have somebody new come on board. We go through many weeks of training so that they're comfortable, we're comfortable, and then of course we're always there to help walk them through anything if need be, if we happen to not be in the building. We also train other institutes. They come in and it's everything from your animal care staff to your PI, your research staff, anybody and everybody, because it really is important that everybody is on the same page with these guys, especially as you're learning about them. And your IACUC may not even know, they may have never heard of a tree shrew. So we really try to work together and figure out what's best for these guys, because they don't all act the same. They don't respond to the same things in the same manner, just as they don't respond to the same people in the same way. Like I said, if we have somebody new that's in the room, you definitely will see a big change in their behavior. So it really is a learning process from beginning to end. And as I've got there, they can't communicate that we do, but they really do have a wealth of information that they're going to share with you. You just really have to slow down long enough to pay attention and really, really become one with your animals, so to speak. So for a health and wellness program, that's important, of course, for any species. Learning what is within normal limits for these guys is important. Like I said, the obvious things you're going to look for, but you're going to try to start to understand what each individual animal's characteristics are. What is their own personality and their behavior? Because that's going to be a tremendous part of understanding what that animal is trying to tell you, whether it's they're happy, they're depressed, or maybe they're not feeling well. It may not be anything serious, but it might be. And you really have to be able to start judging these things. And this is where it's really important that your animal care staff, your veterinary staff, your research staff, everybody is working together. Because of course, you all can't be there at the same moment of every day watching the animal 24/7. So as long as you've got those key components and everybody is trained to really recognize even the smallest little hint of maybe a problem that's going to start, or just they're off their mark for the day, it really is beneficial, because that's those pieces of the puzzle that you try to pull through and work together if there is something that comes up. So an acceptable timeline for your health checks. We do not catch these animals weekly, monthly. We catch them when we absolutely have to. We do everything visually. Again, we're checking for the feces, the urine, their interaction, food intake, water intake, but to just arbitrarily go in and catch them all the time to get a weight or anything like that is extremely stressful for them. Even the animals that do come over to you readily, sometimes, it can cause a great deal of stress. So you kind of have to weigh the odds. The problem with that too is there's just times that you have to catch the animal and you just can't avoid it, because a lot of times they move around so quickly. So a lot of times you really can't get a true assessment of what might be going on health-wise if you don't have your hands on the actual animal. So because of that, we don't routinely do blood work, urinalysis, things like that. Again, it's just so much stress. We really have to rely on our eyes and our ears to go with what we're observing in the room itself. Now, certainly if we have an animal that there's a concern, we're going to do everything that we can clinically to hopefully figure out what the problem is and how we can improve on it. We are working on a system right now. We've got a lot of blood work that we've run, and we're hoping to come up with just some nice basic normals for these guys, from everything from juveniles all the way up to adults and even senior adults. The potential pathogens as well, that's really difficult. We'll get into obviously the COVID-19 later on in this, but we have no idea. So the biggest thing that we go by is we always are trying to be better, safe than sorry. So we're going to take anybody that comes to work that might not be feeling really well, or a cold or something like that, that we would assume is pretty minimal, because of the fact that we don't know if these guys can get it. we're always going to be more careful. Either they're not going to go into the room at all, but if it's absolutely necessary, then they're definitely going to have a mask on, because again, we don't want these guys to get something, and especially if we're not expecting it. So another thing that comes up is your health concerns, injuries, things like that. Again, I always will go back and reflect on assessing your benefit of figuring out what's going on, or potentially a really high level of stress. It's always going to be a fine line that you're going to walk, but sometimes you just have to for the betterment of the animal. As far as medications go, we've had good results, knock on wood, with your general drugs, Baytril for your antibiotic, Medicam, whether that might be a post-operative method or for arthritis in some of your older pets, anything like that. Anesthesia can be a little difficult with these guys, but we've really honed it down over the years and really kind of refined that process. Some of the bigger things that we do see, of course, is managing broken bones, fight wounds, things like that. You can imagine that with the way that tree shrews move around, how it's very difficult to manage a broken bone, if it's something you want to even attempt at all, depending on where it is and how it happened. The fight wounds, that unfortunately can happen relatively often. This could be animals that are just hitting sexual maturity around 4 months of age. It could be animals that have been together for years. They could be siblings. They could be breeding companions. It really, we have not been able to definitively come up with what ticks that cage mate off, so to speak. And it, of course, can be enhanced by different people in the rooms, different stressors that are going on, your HVAC system going down as well. Dystocia is another one that can obviously be a problem, or having difficulty giving birth. In this, again, it can be a wide variety. It could be a new mom. It could be a mom that's in the middle of her breeding cycle, meaning she's had a few litters, she's still got a few to go. It could be one that's at the very end of her breeding cycle. So it's not a surefire answer, unfortunately, that we've come up with, but we do know that stress and things like that obviously can influence it. Depending on, again, where we're at with that animal, where it could be toward the end of their breeding cycle, maybe you do want to look at the information and see, okay, how much longer do you think we have left? Like, what has the trajectory of all of these previous litters been? Have we had a good success rate? Really take a look at the information, just as you would a lot of other species. The other end of it is, maybe it's a breeder that has done a great job. It's had a couple of litters. We have no reason why. So we prep it for surgery. We go in, we do a C-section. We come back out, recover the animal, and eventually return her to the breeding program, and they'll go on and have numerous other litters without a problem in the world. Just another example kind of how you have to stay on your toes with these guys. Managing a broken heart. So, as I said, there are a lot of cage mates that they get along just fine for their whole life. There are others that somebody says something that ticks somebody off, and then boom, here you go, now you've got a problem. But probably my favorite story to tell, and I always tell this story because it's just, as you can tell, we love our little guys. But we had a breeding pair. It was one of our first ones that actually was successful at raising their own babies. And they were just a match made in heaven. So unfortunately, the female was very, very pregnant. She unfortunately broke her leg. And we very quickly started to figure out, how are we going to deal with this? We have a broken bone, a broken limb on a female breeding shrew that's very pregnant, that gets along tremendously with her cage mate. How in the world are we going to even try this, or should we? So we decided, yes, we're going to give it a whirl. We've got to at some point, so why not now? So we went ahead and we put her under anesthesia so we could, palpate the break and everything, kind of get a good, clear picture of what was going on. And we just went old school. We broke up a tongue depressor and we splinted that leg as best as possible, wrapped it up. And then we had to figure out, after recovering her, it's like, okay, now what about this litter, which we knew it was probably not going to be viable, but still, she's pregnant. So at some point, babies are going to come. And it was within a couple of days, actually. Our bigger thing, too, is how in the world are we going to keep these two animals happy that now cannot be in the cage together, because we really have to monitor her well-being. So we scooted a cage right up next to their home cage, and we put her in one and him in the other. And we ended up keeping them there. We were checking on her multiple times throughout the process. And we came in one morning, and believe it or not, these two little kids were reaching their paws through the mesh of the cage, and they were actually touching each other, just being happy, just touching each other, because of course, again, they couldn't interact. Well, let me tell you what, there wasn't a dry eye in the house. That was just so precious to us, and really speaks to how bonded these animals can get. So anyway, her leg healed beautifully. It was about 6 to 8 weeks. We got her back with her mate. They went on to have many, many other litters and lived happily ever after. So that's probably our best story of all, I think. So moving on, diet. Of course, diet is very, very important for all kinds of reasons. If you're looking at, like, when we first got the shrews, their base diet was unsweetened applesauce. So of course, having, for those three of us, the main key players in this, which had that zoo, wildlife, veterinary background, we were floored because we just couldn't believe that we didn't have metabolic bone disease, anything like that, or rickets, any of those things that go along with the lack of nutrition. Well, of course, within a few months, we ended up having our first case that definitely was rickets. We had those fractures in the long bones, the sternum, things like that. So it was no doubt in our mind, if we had any question before, that we had to make a change. We ended up going with the Missouri insectivore diet, and we get it in the granules, which you can see there on the right. And we just soak it every morning. That's their base diet. And then again, they've got the high quality cat food on top, and then their array and rotation, depending on what day of the week it is, to have their fruits, vegetables, bugs, all that kind of good stuff. We do give them the super mealworms, and as you can see from that picture, probably there's a few of you right now that don't like bugs and are getting a little squeamish, but shrews love them. That is the true way to their heart. So anyway, we went through this process, and we figured, how in the world are we going to do this with the colony? Because of course you can't change everything all at once with everyone. You have to pick your subgroup of animals. So as much as we don't want to have singly housed animals, and as social as they are, really the only way to judge something and do your best at this would be to have a singly housed animal. So you can monitor, again, the weight, food, water, feces, urine, things like that. So we took a group of animals that were already singly housed. We got all our basic parameters, and we just went through the process of gradually switching them over. Initially, less of the Missouri insectivore diet was offered, until gradually, a couple of weeks down the road, we had them completely changed over. As far as results, nothing like this happens overnight. You have years and years and years of animals that have had this as their base diet. So it does take time. I would say within a good year, we really had no other episodes, so to speak, or presentations of anything that would give us a concern that there might be a metabolic bone disease or rickets or anything like that. A stray one here and there, yes. And let me back up for a minute. It's not that we had, 10 animals, 20 animals, or anything like that. We had a small few, but again, we just really didn't know how long this was going to take. The other thing that we ended up doing, because for our behavioral program, we actually have worked with Lab Diet to formulate a version of this insectivore diet where it's a pelleted form. We worked back and forth with them to test different ones with different amounts of gelatin in them for a couple of different reasons. Well, number one, we didn't want to change the texture or the flavor or anything like that for the animals, because yet again, we're adding the same type of diet, but just a different presentation of it. So we wanted to make sure that there was hopefully as little waste as possible, because it's not cheap, but also so that it would be more palatable for them. Luckily, it's worked well. We use that for the behavioral animals, like I said, because we do sometimes have to restrict their water intake to help motivate them to do their trials. So we've had good results so far, and hopefully it continues. So the previous caging that we had when we first got them at MPFI, you'll see on that left side, it was actually, those are metal primate cages. That's what our juveniles were in. They were heavy, loud, but they worked obviously for that time period until we could come up with something different. The cages on the right that you see, obviously, I'm sure a few of you are cringing right now. They needed some work, but again, at that time, we could only change one thing, at a time, and we really had to be careful about that. We didn't want to put too much undue stress on these guys. So housing for all ages, what you've got in-house may actually work. Our Tecniplast cages that you see here, that's what we have for our rodents. So it was a matter of how can we kind of modify these to work for the shrews when they're in those infant and juvenile stages before they go to a larger cage. So you've got the baby bucket on the left, and yes, for those of you wondering, that is food takeout containers, but they work great. And we've got that. Then we've got, of course, the IBC cage there in the middle. That's what they'll go into when they first come out of the incubator, and then the double decker right before they would go into an adult housing situation. This is the incubator for the newborns. Very simple. It's a wonderful product. Got a little shelf there because we do have surges of babies, obviously, that come in, so that gives us a little extra space. In the bucket, you'll see that number on the top. That's breeding tree shrew cage, and that's the cage number. And then chem wipes we have in there to keep the little guys comfortable. They'll stay in here about 3 weeks until their eyes open. Then, as I said, they're going to go to an IBC cage. So initially, when we bring them out of the incubator, especially if it's a singleton, if it's a singly housed animal, which we will, eventually, before they move out of the infant room, we'll go ahead and put animals together, males together, females together, just so that they can continue to grow up and hopefully get along just fine. But they're going to initially be on a water circulating heating pad or a heating blanket, just so we can make sure they can thermoregulate. And then we're going to switch them over to the ventilated rack, just like you would for a rodent. So I don't know if you can see really clearly there in that picture on the left with the cage cards and everything, but there is a green stump that's in there, and that's what the little guys will hang out in. Probably for at least that first week, they're not going to come out of that at all, except when you come in once a day to feed them. Then they're going to just sleep, and that's really going to be it. Eventually, then you start seeing the brave ones come out and start wandering around and sniffing at the food and the toys and different things like that. They're going to stay in these cages here until they're completely weaned from milk and they are eating the insectivore diet, the toppings, and they're really growing and thriving. Then, like I said, the double decker, that's also on a ventilated rack that they're housed. This is the time period before they're going to go to an adult cage and be introduced to the rest of the colony and all those crazy noises and, smells and things that go along with it. They've got a shelf in there. For the mesh itself, pretty much for both cages, the IBC and for this one, all we did was take flexible mesh and then non-toxic, high-temp silicone so we could adhere it to there, because we really wanted these stages for the little guys to be exploring, interacting with each other, climbing, getting those legs stronger, getting ready for those big cages. And then we have the crowning glory, the adult cage. So this was really a godsend for us, because as you remember the caging before, we needed some help with this, but we needed to come up with something that was really good for the animals, but also easy for us, so that as we needed to work with them and maneuver them around, it was hopefully as easily as possible. So the picture on your left is a cage bank, top cage, bottom cage there. You have 4 ports for nesting tubes. You don't have to use nesting tubes on every single port. There's a little metal cap that comes in there with it. And they actually really like that cap. A lot of them will sleep in there, just hang out, pick on their cage mate, whatever they want to do. So you've got that option, especially if you've got, a breeding pair that are raising their own, then you've got another option for the babies to be in and mom and dad to be in another spot. You've got a resting shelf there, toys, manzanita branch. We've got crinkle paper there on the bottom of the cage. So whenever cages are being changed, we spray essential oils, whether it's lavender or basil, anything like that. And it's okay, Vera, sorry. And they literally surf around with that little scent gland. They're rubbing all over everything. And from everything that we can tell, they're really having a good time, but we always want to keep a close eye out and make sure, even this is not something that's really stressful for them. Maybe even at some point testing out the idea of, instead of changing the entire cage bank out every 2 weeks, maybe just, keep some of the toys in there, the manzanita branch or something like that. We're always looking for those different ideas. You've also got the food door, of course, so you don't have to open the main door, which is great for not just giving them their diet each day, but also interacting with them, assisting in catching them. giving them enrichment in the afternoons, anything at all. So these were two really big components for us when we were working with alternative design, because again, we want it to be easy and efficient. The back coming forward, to have that squeeze back, was really important because every animal doesn't want to interact with us. They really couldn't care less. For those, it does help to have that back come forward. The other thing was, well, what about changing cages or introducing new animals or anything like that? So we literally just took the ends off of those nesting tubes and take a fern coat, like for plumbing, and we joined the two cages together. Put the casters in lock because you don't want any extra movement going on if you can avoid it. And then there's guillotine doors that come down. I don't really know that you can see it clearly in this picture, but there's one at the cage side right there against the mesh. And then there's another slot that's a couple of inches over. That was big for us in the design of this, because we also wanted to be able to use these nesting tubes as transport tubes if we needed to. So you can literally have an animal in there, close the tube off, remove it successfully without then obviously having any other animal that might be in the cage get out. And it really has done well. So enrichment, we have a huge conglomeration of things that we give them. Scent enrichment, of course, toys, all kinds of things like that. Aquarium plants, you can see on the left, they love those, scent marking them, rolling around. We're never going to take the place, again, for the animals that do want to socialize with us and are curious about us, to have that connection with them. And then, of course, the little guys at the bottom, like I said, scooting around, learning how to climb and really navigate in the cage. Something really cool that happened, like I said, there's all kind of toys. That's basically in the middle a cat toy. For those of you that aren't familiar with what that paper looking product is, that is a diamond twist. So it's literally taking a paper towel and you're just twisting it mechanically as hard as possible so that it stays in that form. And we take them and actually put them in Ziploc bags so that we can put, whether it's dill in there or maybe cinnamon, sage, anything at all. And so it's twofold for the enrichment. You've got the diamond twist itself to play with, but then you've also got the scent side of it. So these we intertwine throughout the mesh of the cage. The shrews love it. They can rub on it. They can pull at it, play with it, do whatever they want. But we figured out early on that we have some really smart guys that finally got the diamond twist out of the mesh. And then they decided they were going to weave it in and out of those little bars of that toy, which we were pleasantly shocked, stunned, and amazed, because obviously that speaks to that higher intelligence level that they do have. So these are some videos, and bear with me because it's a little bit of background noise, but that's a combination of our HVAC, some of us talking, and vocalization from the colony itself. And because all of this is new as far as the Zoom thing with getting the videos to work, we'll give it our best shot. So this is fresh dill. Oops, sorry. Like I said, this is a challenge. And you can see she's using that scent gland to rub on there and to really scent mark it and play with it and have a great time. And we've got Jello, which is twofold. You've got the actual item itself that tastes good. It's wiggly, it's fun to play with, and you've got the cup itself as well. You can tell these two guys are really making sure they're comfortable, but they're making sure that we're not doing anything crazy. And they'll eventually go through and start scent marking it and having a grand time. Then we've got an aquarium plant, and this is literally what we did the first round, is we just took it and set it on the shelf so we could see what their response was actually going to be. If I can find my mouse. That's Natasha. She had some really big ears that stuck out, but she was a sweetheart. You'll see her start actually mouthing it to test the texture of it, scent marking. And so she's really comfortable with it. Like I said, it's on the shelf, so she'll do a little slip. There we go. And then this is a part, this is a little blurry at the beginning, but this is some juveniles. So after they've had their milk, they're in that IVC cage, and we really try, when there's animals that are roughly the same age, we really want them to have that interaction, you know, with other animals, their own species, not just us. It's not to tame them, it's just to make them as comfortable as possible when we do have to work with them. So these little guys, you'll see, are and you can see they're already starting to learn that scent marking behavior, and this is about 6 to 8 weeks of age. There's nothing better than this age, I'll tell you that right now. All right, now that we got finished with those fun videos. So breeding. Breeding is a whole different ballgame. We actually have some institutes that we collaborate with that they don't have any desire really to start a breeding program. You know, they do realize there's a lot more work involved with it. There's a lot more things that you need to watch for. It's kind of a different beast at times, so to speak. So that's one question, because there's plenty of them that don't have a breeding colony. There's others that do that. This is another one of those things that will, you know, if you come in and train, or even if it's over the phone, whatever help we can give with our collaborators, we do, so that we can, again, try to improve the well-being for these little guys. So we don't have, as far as who are you going to breed, we don't have Charles River or Jax or anybody else that's giving us, you know, this cool little program and here's this animal and this animal. Sure, go ahead, breed them. We don't have that. So our colony is decades old. All of the information that we have, we end up basically looking at every single, like every piece of it. And we go with the animals that we have that are the farthest apart as far as, you know, any relatives or anything like that. If there are animals that you do end up getting from us, this is another thing that we'll work with, and you decide later down the road, let's say that you want to breed them, then we can work with you on, okay, who is really compatible, and help walk you through it again. Because remember, when we're training other places, it sometimes can be really close to a year by the time we work with alternative design for cage, or whoever you choose. You know, if you do need to order caging, we went with alternative design, obviously. So giving them the time to actually make those cages for you, anything at all, and walk you through the process. Then you've got, of course, a couple of different options. Parent rearing. We are never going to get better than mom and dad. We have found that with our animals that are parent raised, we see almost every single one of them, both the male and the female, take a large role in taking care of the animals, which is pretty ironic, considering the fact that in the wild, mom only comes to that nest every 24 to 48 hours. She has only seen them by the time they're 3 weeks old and their eyes start to open. She's only been around them a handful of minutes, you know, all this time. So that's something that we had to remember. Parent rearing is wonderful. That's the best thing in the world. But your cage dynamics, your room dynamics, all of those things come into play, because we're really going against what these animals are built to do, is to have their offspring right there with them in the same area. For those that don't raise their own, we do hand rearing. We had a little bit of a challenge early on when we first got the babies. The success rate for raising the infants was 37%, which was shocking. You know, there were enough animals for research, but again, you kick back into that zoo, wildlife, vet tech mode, and it's like, what are you talking about? 37%, this is crazy. So again, we had another thing that we knew we had to work on. As far as the infant formula and everything, we really went through some trial and error. We had our subset of animals, just like we did for changing the adults over to the insectivore diet. And we finally came up with a formulation pooling from all of the knowledge that the three of us had. And we said, okay, mom's milk is 25% fat. We need the protein, we need that fat, because they really hit these crazy milestones as they're growing and developing. And we need to have something that we can really start to see the results on. So we ended up with the mixture of an egg yolk, heavy whipping cream, and kitten replacement milk, which, knock on wood, we've been at 100% success rate for a few years, not to jinx myself. Breeding does occur throughout the year here, because our vivariums, for the most part in the US, don't have windows, or not where the animals are housed at least. In China, they only have breeding half the year because they do have windows in their vivariums. So they depend heavily on the seasonal changes. Their gestation period is going to be around 45 days. That's an excellent breeder if you have a litter every 45 days. 1 to 5 infants, that's your span, I would say. You'll see one infant if it's a mom really early on in her breeding, potentially, but you'll really start to see that as they're coming off that back slope and on the back end of that breeding cycle. Normal is going to be 2 to 3. When we first got them, I think I found one sighting where they had 4 babies. So when we finally had 4 babies, we were high-fiving. We're like, oh yeah, this is awesome. We've got happy animals. We've got healthy animals. This is great. Well, then lo and behold, we had got 5 animals, 5 babies that nobody had heard of before at all, anywhere. And it was not, don't get me wrong, we were excited, but I remember saying to the other technician, I'm like, you know what, just go back and check. Maybe you counted wrong. Maybe you counted somebody twice. Lo and behold, she comes back and she's like, nope, we got 5. We had 5 healthy babies, not a runt in the bunch. Everybody was fed, they were warm, they were happy, and they obviously ended up growing up with no problems whatsoever. So that's like the core of the general husbandry and things like that. So obviously, like I said, this can be a roller coaster. So you can get pretty frustrated at times because anything that you bring to the table, you know, your knowledge is good, but everything that makes sense with other species, for the most part, doesn't make sense with these guys. So you really kind of have to work together to figure things out. But then on the flip side of it, you've got the science and you've got the caring of the animals and you've got then collaborators at other institutes that have them. And then you realize, oh my gosh, but look at how many things that we've actually learned. So then we step forward to the science and what we utilize them for at Max Planck. So again, remember, we're talking about years ago, they had this question, you know, are they squirrels? Are they primates? Are they rodents? What in the world's going on? And we really started to see that the primitive primate characteristics came into play because they do have a highly developed brain. So we look at the visual system, we see how, you know, it functions, the organization and all that fun stuff. So their brain there that's on the left in that picture, that cortical structure allows for a much higher brain function, like the social, emotional, learning memory, things like that. So they're in that center picture. So their visual cortex contains columnar organization. So meaning like if you could take and just put a column straight down through that visual cortex, you're actually going to see the neurons within that column have a similar response to different types of stimuli or things that we show them or present to them. And then from there, we can create these really cool orientation maps, which is the picture on the right. So when we're talking about an orientation map or orientation tuning or things like that, we're talking about how a cell is responding strongly to some things that we show them and then poorly to others. And then we make those maps. Overall, those orientation preferences that they have that these cells have can change continuously, but for the most part, they're staying within that parameter. So we're really looking for how the cells within these maps and in these columns can then represent, you know, things in their world, like a branch, different stimuli, and things to help them navigate. So you'll see here, I've highlighted the top left, the primary visual cortex, which is the dorsal surface of each hemisphere. That bottom left picture shows other cortical areas that have been discovered, which are highlighted in blue. And then the picture on the right is actually our histology. It's a coronal slice, so anterior to posterior, front to back, of the visual cortex that's actually been stained to show just how dense these cells are at all the different layers throughout that process. So one of the ways, other than just observing their general behavior and attitude and interacting with us and how curious they are, is this task. So this is a go, no go, where basically they are shown their target or what we want to show them, the correct stimuli, so to speak, that they're going to get a reward for when they see it. So then they're shown an incorrect stimuli. So if they try to get a reward for that one, they're going to have a timeout. But if they don't actually respond to that and try to get a reward, after the after the trial resets, I guess is a better way to put it, then they're actually going to get their reward. So in their discrimination of different stimuli and different things we show them, they can actually judge changes in things that we're giving them down to about 2 to 2.5 degrees off of their target, which is crazy. Kind of to correlate it, humans and primates can get it down to about 1 to 2 degrees. So this really is an example of how closely related they are, you know, with their higher brain functions. So beginning your research and, you know, hoping that it flourishes. Again, I keep just repeating the fact that it's so important that everybody, whether it's an animal care staff, your veterinary staff, your researchers, anybody at all that is going to be in contact all the time with these animals, really builds a relationship. The last thing you want to do is come in and the only time you're asking an animal to do anything is when you're asking them to really only do what you need to do, like they're not getting anything out of it. So you want to build that bond with them. This is Lolita on the left there. She was a trooper and a pistol at times, but she was a good girl. Anyway, she was very early on into our behavioral program, and she was great. We learned an awful lot of many things from her, not just from the science end, but also from interacting and the husbandry end. Again, that training is super important because you're wanting, if it's an animal technician, if it's a researcher, anybody to be on the same page. These animals hide things so well sometimes, you know, how they're feeling, if they're starting to not feel well, that they're very subtle changes. So if everybody is on the same page, you have a much less, you know, chance to actually miss something if you, hopefully can avoid it. So in the beginning, I literally sat for hours and hours and hours and weeks and weeks and weeks with many notebooks, and we needed to learn. We needed to learn about these guys. We needed to see what their capabilities were of their own free will and not just things that we thought down the road we might ask them to do. As you can see, there's a hamster wheel in that double decker. We didn't know, so I'm like, sure, let's go ahead, toss it in there. Let's see what happens. You know, it was just stuff like that. Our end goal, of course, with our research project, with our behavioral end of things, is to have awake imaging. So you'll see there on the right, it literally started early on with me trying to figure out, okay, what kind of rewards do I have that these guys are going to be interested in? What is going to work well in a syringe pump? What is going to keep them in a tube for as long as possible, focused on one stimulus, and how long can I actually keep them in there and then be comfortable? Because that's the goal, is to keep them comfortable with what's going on. So I literally took a little Rubbermaid container there, drilled some holes in it, had the tube in the side from the syringe pump with their reward, taped a PVC tube in the bottom, and I would just let them explore it and go through all these different phases until finally I figured out, after, gosh, probably 30 different items that I was trying with them and different mixtures, until I finally figured out, okay, they really love apple juice, unsweetened apple juice, and that would hold their attention. They don't fill up on it really quickly, and they literally would stay in there sometimes for 15 to 20 minutes, which is great, because for awake imaging, that's what we need. We need them to be focused on their stimulus and being able to accept their rewards. So throughout the process of your research projects, and this is stuff you guys all know, talk about what those questions are you're trying to answer. Revisit it really often, because it is a long road, and especially if it's a species that, there's just not a wealth of information about, you really have to communicate a lot and figure out the best way to go. And there's no better way than to have all of those people that are involved to have a say in it. They're validated, and your research is definitely going to benefit from it. All right, so the changes that we have made to cope with this pandemic. So one thing, of course, and we all know this, so navigating employee scheduling to give our animals the best care possible. They can obviously feel if we're stressed or we're tense, and they pick up on a lot of these things. So your scheduling of your employees, what we've done is we have two sets. We have one team, so to speak, that goes in one week and one team that goes in the next. That works out well because, again, you've got to have that continuity. Everything needs to flow. You want, of course, the animals' physical and mental well-being to be enhanced, but also for all of us. We saw, early on with our guys that especially the animals that were much more social and interested in communicating with us, so to speak, and definitely our behavioral animals, that they were much more quiet. They appeared to be a little depressed. So we really kind of reevaluated a little bit early on in this, and we were like, okay, so we've got the regular group that comes in and takes care of them throughout the day. And at the end of the day, myself and another person goes in and we just give them another opportunity to interact and to have another piece of enrichment item. And, and of course, again, check on everybody to make sure they're doing well. But it's always good throughout this process because this is difficult on all of us for many different reasons. But communication is key. You really have to work with the teams. And I'm not talking if you're, a veterinarian or IACUC or an animal care technician, a PI, whatever it is. It's important for all of us to communicate. This is the time that you see all those things that maybe you've wanted to work on or you didn't have time to work on. Sometimes these things start to surface. So don't be afraid, through this, but especially afterwards, talk to each other, sit down, have an open discussion about what was all of our experience, with this, because it's probably going to be a little different across the board. Let's figure out how we can work on things and continue to give ourselves and of course the animals the best care that we possibly can. Our research for now has obviously stopped. We're just focused right now on humans and animals all remaining safe and well fed and cared for. Our PPE before this was obviously gloves and dedicated either scrubs or isolation gown there that you can see. Since this started though, like I said, we have no idea what these guys can get. We know now that we can be carriers of COVID-19 and not exhibit any symptoms at all. So what bigger fear could we possibly have than our animals that we are working with every day and that we love really catching this? And then we have no way to know it, but then they're potentially spreading it to other animals or to us or vice versa. So we'll eventually get back to that, once our research starts back up. But for now, we definitely, of course, again, the gown, the gloves, must wear a mask. Prior to this, we use chlorhexidine as our disinfectant because it's, been around for years. It's a great disinfectant for many different things, but it's also benign enough so that if the animal comes into contact with it, we're not, really concerned about them touching it. For all intents and purposes and all the testing that's been done, it's safe. But what we have changed is to go to the peroxyguard because that is a little more high powered, especially for non-porous surfaces. So again, we don't have animals going back and forth right now in transport tubes, but if for some reason we did have to catch one and really, assess them and put them in the tube to get their weight and all of that, we still would go ahead and definitely use the peroxyguard, but then maybe follow up with that chlorhexidine to get any residual out and really get them, kind of as safe as we can possibly be, hopefully, and not have any types of problems with the disinfectants. Testing, we have gotten wind that there is some testing, not COVID-19 testing, but testing to see exactly what forms of the coronavirus that they can get in China. We don't have any more information than that right now. We're hoping that that's true because anything that we can possibly find out to help us with these little guys is amazing, but right now it's, it really would be critical. Challenges moving forward, it's just going to be for all of us to get back to a sense of normalcy eventually, get back with our animals and the people that we love as well, and and hopefully find a new normal for all of us. So anyway, with that, big thank you again to our sponsors before we actually get to any questions or anything like that. And I will toss it back to Austin.
Thanks, Ms. Freling. We've got a handful of questions from the audience. I'm going to begin delivering them to you. First question, Ms. Freling, do you always pair or multi-house tree shrews?
We always start out with housing no more than 3 together, especially as they're juveniles, just to give them plenty of room. But they are social animals, so we always want them to have a cage mate if possible. Sometimes, it doesn't work out that way in the long term, but yes, they are always with another animal if we can.
Ms. Freling, have you ever tried positive reinforcement training to help with exams or other things?
Definitely. We use multiple things such as dried mealworms, things like that. And for some it does work, especially those that are a little more social. But again, even those that are social, you're asking them to kind of be confined, be really close to us. But yes, we do. And any suggestions anybody has, hey, bring them on.
Here's the next question, Ms. Freling. How long is the breeding cycle, and at what age do you expect they can no longer produce litters?
Well, that's interesting because we've seen quite a variety. We've had them together as early as 6 months of age. We've had them together, put together at 2 years of age. We've had animals, females that are 4 years old, continuing to have litters that seem to be healthy. And then we've had others that, you know, they have litters literally every 45 days like clockwork. And then all of a sudden, you know, they just start to dwindle off. But we, at our peak for breeding, our females, you're going to see them are really good breeders. You're going to see them have 20, 23, 24, 25 litters.
Here's the next question, Ms. Freling. Are the newborns kept with the mother, or are they alone in the incubator and IVC cages?
If the parents will be raising them themselves with no contact other than visual contact from us, obviously they're going to stay in the tube, in the nesting tube, in the cage with them. For those that don't raise their own, they are pulled the morning that they're born, and then they're housed in the incubator with the other litters, you know, in their individual boxes until about 3 weeks when they move into those IVCs.
Here's the next question. Is it preferable to have solid panel sides on caging so breeders do not see other tree shrews?
You know, that's not a clear answer. I think that it definitely wouldn't hurt. We don't have them on ours. That is something to consider. Again, you've got so much that's influencing everything. You could have them on one, you know, have a panel on one side, or you could have a panel on both, because every animal, for the most part, has a cage on either side of them. So another thing to remember, though, is as you're blocking them from seeing other things, you're also narrowing your ability to really visualize that cage, especially if there's a health concern or a new litter born that happens to not be in the nesting tube. So I think it can go either way. I think it can work. I think there's just some pros and cons maybe to both.
Were the aquarium plants used as enrichment real plants?
No, not the aquarium plants themselves that you saw on the shelf there that Natasha was scent marking. We do have, you know, ourselves that work there. If we have rosemary or dill or anything like that, that we do not use any chemicals on whatsoever, and we know it for a fact, then that we will bring in for them to interact with.
Do both males and females scent mark?
Yes, they do. You know, as with a lot of things, though, we have plenty of females that don't necessarily keep themselves very tidy, so to speak. But you will see a fair number of, especially those larger, older males, that scent gland just gets, it's nasty. But yeah, they both, to answer your question.
Here's the next question, Ms. Freling. Is there any enrichment or food you would not recommend to give them?
The one thing I definitely would tell you not to give is citrus. Because they do scent mark things so much, we don't want there to be any burns or anything like that, you know, their neck or their chest or anything. So we really don't give that at all, because some of them will just scent mark the heck out of something. So we really steer clear of that. We always just keep in mind with any enrichment item, whether it's food, toy, or otherwise, you know, what could they destroy and what can they actually maybe ingest, because we don't want to go down that road unless we have to.
Ms. Freling, where did you get your original animals?
Our original animals were actually part of a colony that divided. They were at Duke University, and one group went to UAB, and then our PI, Dr. Fitzpatrick, he's actually had this colony. So that's where they were just prior, was at Duke, and then they came to us.
You said the tree shrews were good parents, so why do they or do they not feed their young?
You know, that's something that we've really tried to figure out, is why does it work for some and not for others? The biggest reason that we believe is they're just not wired that way. You know, they're not wired to have their infants right there with them all the time. That's the way that Mother Nature made them. So as mom's not going to be there for every, you know, except every 24 to 48 hours, and they engorge themselves on milk to carry them throughout that time, that's really the only thing. Again, if you're only taking it from a standpoint of those particular breeders, ignoring any stressors that could be going on in the housing room or, you know, other cages in the room, so to speak.
Ms. Freling, what is the incidence rate of bone fractures in the colony, and at what age do they tend to be most frequent?
We've had them at a variety of ages. I would say most likely, you know, the older ones, of course, they're maneuvering around a lot faster. We have not had many. Oh, I'm going to jinx myself on this, but we have not. We have had, if memory serves me correctly, probably about 4. But 4's too many, you know, if it's something that we can avoid or try to figure out. So it really was not many, but that was too many for us.
Is the incidence of bone fracture related at all to the tree shrew diet?
That is a good question, because early on, you know, that was a huge component, and I would be foolish to sit here and say that that did not have some kind of influence on it.
Here's the next question, Ms. Freling. Have you had to import any new animals to increase genetic diversity?
That is a goal of ours, and actually we have been in the process of doing that. Unfortunately, with everything that's gone on, that has come to a screeching halt. And obviously, since that area of the world is where this problem originated, you know, we really don't know when we're going to be able to do that, but that is definitely still our goal, because as I said, the colony has been in existence for many, many years, for decades, and those tree branches get tighter and tighter. So yes, we definitely plan to get other shrews.
Ms. Freling, when you first started the colony, did you have issues with cage sanitation and residual cleaning chemicals causing stress, knowing that tree shrews are scent markers?
We really didn't. Nothing that we saw. But we didn't have any residual or anything like that. Now, I will say this. So the double-decker cages and the IVC cage, how they have the slot for the water bottles, you know, you may see some debris in those where it can pool, you know, until you get it out of the cage wash equipment. But generally speaking, no, there was no residual or anything like that that we had to deal with, thank goodness.
Ms. Freeling, have you tried clicker training in addition to their liquid treat?
Yes, I did early on. It worked for some. It didn't necessarily work for others. But I did have some decent results with it. But when we figured out that it was going to be a little easier to figure out what we could present them that would keep them really interested, and then couple that, of course, with if we needed to do water restriction, that really seemed to work better. But yes, we did try clicker training.
Do you find the hand-raised babies become equally nippy?
Oh, yes. And actually, I will say this, that's a really good question. It really doesn't matter whether they're parent reared or hand reared. We see a huge conglomeration, just a mixture of different attitudes, personalities, however you want to put it. And we don't necessarily see that hand reared babies or parent reared babies are better animals for me to pull into the behavioral program. We can have an animal that is from a parent that is the best thing in the world, and then we will have another one that literally looks like he wants to kill you. So unfortunately, we haven't found that. Otherwise, my job would be a lot easier on picking animals for the behavior side.
Ms. Freeling, what makes tree shrews a better model in your studies than mice or rats?
Because of that higher brain function, honestly. And rodents, you know, that's a kind of a, what works with this one usually will work with the next, and so forth and so on. These guys, because of their capabilities to learn different things, but also associate them and interpret them in different ways, that really has been, and the way their visual cortex is developed, that's really been a great bonus. And of course, you know, if you're looking at different things with, some people may be working with primates as well, everything is just easier. It's the breeding time, as far as the gestation period, is smaller, it's less expensive. We don't have all of these crazy animal protesters and things like that yet, that are really trying to fight us on using non-human primates. So this is kind of a balance.
Ms. Freling, with the milk replacement heavy cream and egg yolk, have you encountered any signs of diarrhea? And if so, what do you do to stop it?
Yes, thank you. I forgot to mention that. We have found that this new formulation, newer, I guess if you want to put it that way, is awesome. We have a couple of time points throughout those first 3 weeks that we have seen some issues start. We call it diarrhea. You'll have a weird odor to the feces. Sometimes it'll be like a green or a yellow, kind of a gold even. So we tried a couple of other things, but it really just, there was no change whatsoever. And the biggest thing, of course, when you have infants is once you start on a pathway of having health concerns, it is a slippery slope and it usually doesn't take very long. So what we do is we give them Pepto when these situations start. We give them Pepto-Bismol before they eat each day, and that is always, always, always coupled with sub-Q fluids, just to make sure we don't have to start dealing with any dehydration issues or anything like that. From that point then, once we get past around P5 to P7, if we see it then we get to P10, P12, we might see it then, but usually after that, we're pretty good to go. And that doesn't happen very often, but that's what we do whenever something like that starts.
Ms. Freling, what is the optimal temperature range for tree shrews?
That's another good question. When we first got them, we're thinking, okay, Southeast Asia, jungle. Well, these guys, of course, they're built that way, but they don't know that they came from China in the jungle. We keep the rooms basically what you would for rodents. You've got that, 40 to 60% humidity, give or take, and it's around 73 degrees. Of course, there's a range, but that's typically what it's most comfortable at. And the incubator is set for, a little bit higher on the scale for the humidity and the temperature, but in all honesty, it's not that drastic, not what you would think.
Here's the next question. For those cages you showed pictures of, are they able to be connected to automatic waterers, or do they receive water bottles or dishes?
Correct, they receive water bottles. We have not explored any type of a system to attach them to the automatic watering system. And it actually helps you if you're watching an animal for a health concern, or again, if you're in a diet change or anything like that, so you can see exactly how much water intake they've got.
Ms. Freling, what is the general disposition of tree shrews? We're getting some questions from the audience about whether they're friendly or aggressive.
A wide variety, but initially I would say they really don't want to have much to do with you. Some are more curious, like I referred to a few times earlier, but others really couldn't care less. So again, we just want to keep them as comfortable as possible and not stress them out if we don't have to, to catch them or anything like that.
Here's the next question, Ms. Freling. We have someone in the audience has an obese, singly housed female, and they're curious about ideas for weight loss. They gave the animal double cage access for more room to move, but she loves her tube.
Yeah. The one thing that I can recommend, and that's not an odd question, and it's not necessarily that you've overfed the animal or something like that. We can literally have siblings that once they, reach adulthood, one can be much larger than the other. The one thing that I would recommend is you don't want to stress the animal very much again, but try once they're out of that tube, just drop down a guillotine door, or if you don't have alternative design caging, something to block off that tube, just to give her some socialization outside of that space. But again, just kind of from a distance, watch her closely and make sure you don't have a lot of added stress going on because she's digging at that tube trying to get in there.
Ms. Freling, what types of research studies are conducted with tree shrews?
There's quite a variety. Obviously, the visual system is the biggest component. They definitely will, or retina work, anything like that. They will someday be a great model, I think, for different types of cancer, mammary tumors and things like that. We will see, we've had some odd carcinomas and stuff like that. And somebody may already be kind of starting that. in Europe or somewhere like that, but not to my knowledge right now anyway at this point. But I think they also are used for some hepatitis studies. But I think they're going to be a really much larger model than what they already have become.
Ms. Freling, what drugs do you commonly use for sedation?
Well, it depends on what it is. So if we've got an animal that's going to actually go upstairs to have a cranial injection or something like that, then we'll give midazolam initially once we catch them, just to kind of take the edge off. And then they're put into the transport tube. And then once they're upstairs to the surgical suite, then we'll go ahead with ketamine, atropine, buprenorphine SR, things like that. Again, it kind of just depends on what it is. If it's something at the cage level where it's maybe a fight wound that we really need to flush or something like that, a lot of times, again, we're depending on which animal this is and how they're exhibiting whatever problem is going on, then we'll maybe even just mask it down with isoflurane and oxygen. It doesn't always have to be injectables.
Here's the next question. Are tree shrews jumpers at the 3-week age or earlier?
They will start, what's a good way to put this? A jerking type, where they're moving quickly just as far as their stance. But when they really start to try to jump out of the cage, the IVC cage when they're little, that's more likely going to be around that 6-week mark, because they're really starting to stretch those legs and learn their surroundings. And some of them, even in the incubator, the way that they would react with being bottle fed or something like that, you can tell sometimes what their personality is going to be like, even at that young age.
We have a couple more questions, Ms. Freling. Is milk for human infants suitable for infant tree shrews, or do they need special milk?
They definitely need special milk. Mom's milk is 25% fat, and for a high metabolic rate and for the way that they're moving around, even though, not necessarily jumping all over the place at a week old, I'm not saying that, but we really need to have that high fat content and that protein, because you're really going to start seeing them make big leaps as far as their growth spurts. So we really want to be able to support that and really beef them up, so to speak.
Okay, last question, Ms. Freling. What type of behavior do tree shrews exhibit when stressed due to overhandling?
You can see everything from just running all over the cage, like with nowhere to go, like just let me get away kind of thing. You'll also see them just remain very quiet sometimes. Again, we're talking about depending on what the situation may be. Vocalizing, like just screaming sometimes if they're very stressed. For cases like that, then that's when you're going to want to look at really closely and really quickly. Is this an animal that we need to just kind of quickly sedate so we can take the edge off and calm it down, and also, all the other animals to take that stress away from them. But you'll definitely see that if they're stressed. Also, you can see that they're not eating. They seem depressed. So again, it can be a huge variety of things. I really wish it was a blanket statement, or it was if this happens, do this, and if this happens, do that. But it really isn't. It's just an individual based situation with what you're up against with each particular case.
Ms. Freling, thanks very much for your generous time today.
Thank you. Thank you, everybody, for tuning in.
This webinar was sponsored by Animal Care Systems and was in no way endorsed by ALAS. Each webinar in the series is produced independently by each company. Special thanks go to Jay Campbell of Somark Innovations for his continued facilitation of the series. For details about upcoming webinars, visit the ACE community postings and ALAS LinkedIn page. Today's webinar will be freely available in the coming days on the Animal Care Systems website, and keep a lookout later in spring for our next webinar announcement. We hope you all are safe and healthy during this unusual period, and we hope you can keep up the good science.